Hello, my name is Sean, and I will be presenting a reprogrammable CRISPR DCase9 method for epigenome editing. This is a pretty specific method, so we're going to go ahead and refamiliarize ourselves with central dogma real quick, and we'll start in with proteins. Proteins, protein refers to an incredibly diverse group of objects, much like I if I referred to everything on this page as building materials. Certainly bricks and rebar and the heavy machinery I could see, but the land needed surveyed, so you needed a drone or hand tools and power tools. You can kind of see how building materials is stretched, and so too proteins are very diverse. So we're going to narrow that down a little bit to enzymes, and enzymes would exist somewhere over here on the right between the hand tools, power tools, and drones. So enzymes are tools. And just like tools, form fits function with these. Now it's easy to look at a hammer and see what we might apply that towards. Proteins perform strange functions in a strange environment, so it's very hard to just look at them and see what it does. So I've rendered our hammer into a protein type depiction, so that'll serve as our abstract anchor. So importantly, with this hammer, you've got a start point and end point, and it's an unbroken strand all the way through. And that means that this is a single gene product. I mean, that comes back. That's important. So let's take a look at this start point and kind of zoom in a little further here. So we've got our start point right here, and I've got these little different shapes depicting our amino acids. And it works because amino acids really inform the overall structure through their unitary geometry. So if you want a straight line, you can have these. If you want a curve, you're gonna have to have this sequence. And so we can see how each one of those little units goes together to make this whole hammer, and how by programming each unit, you can make a hammer. Another thing you need to know about proteins, active domains. So that, or enzymes, each enzyme will have an active domain. So you can see on a little hammer protein over here, the active domain is where you make contact. On other enzymes, it will just be the area where the action happens. So we're not going to spend too much time on synthesis. I just want you thinking about how DNA informs RNA, informs protein structure. Okay, and got your little RNA and ribosome and everything going on here. So if I wanted to make just a specific, say I just wanted the mallet off of my little protein hammer, what I could do is limit my DNA sequence to just the mallet, and then run that through, and I would just create a mallet. So that also comes back in a little bit. Final thing I want to look at with the DNA is the CPG spot. Between a cytosine and a guanine, there exists an opportunity for a methyl group to be added. And that comes in big right now. So regulation. We can make my little enzyme hammer, but how do we stop making enzyme hammers when we have enough? And protein transcription can be regulated from a DNA level through methylation. You have these certain regions called CPG islands, and they're cytosine guanine rich sections that are amenable to a bunch of methyl groups being added. And when those methyl groups are sitting in the DNA, they provide steric hindrance to any polymerase that wants to get in there and transcribe some RNA. So it effectively shuts down protein synthesis. On the contrary, in a demethylated situation, everything's very open, very easy to bind a polymerase. So that is a wide open situation. And that takes us into epigenetics. Epigenetics studies this regulation, and we've got a little chemical breakdown of the TET1 demethylation up here and DNMT methylation down here. What we need to know for this presentation, TET1 creates this bottom situation, demethylation. DNMT3 creates the top situation, methylated and closed down. So we're going to stick a pin in epigenetics real quick and talk about CRISPR. So CRISPR Cas9 is an enzyme that was originally a protoimmune system for bacteria, and it essentially functioned as a locus programmable drone with a set of scissors, and it would function to find enemy DNA or foreign DNA and snip it to bits. So genetic researchers have co-opted this capability from many different things. For my application, I use CRISPR Cas9, which it's that same, it's got the drone functionality, you can program the location, but it has no scissors. So what can I do with this enzyme? And the answer is anything that I program it to do. So in reality, we programmed our Cas9 with a DNMT3A active site or active domain. So how did we do this, or how do we do this? First, you have to identify the active domain, and you do that through protein alignment. Then you design primers to amplify just that section. You just want that little piece of your protein. So you just need that sequence. Then you electrophorese and purify, verify that base pair count. And then you're going to go ahead and ligate. We ligated into a PGMT plasmid. We transformed and then plated on IPTG and XGAL. We grabbed our little white colonies and we extracted, purified, and digested. Down here you can see a depiction of our active domain insert. We then took that insert, we ligated into a CRISPR plasmid. We sent that CRISPR plasmid in to verify that we indeed didn't frame shift or anything, that we had an active enzyme, and then we injected. And all you really have to pay attention to in the injection is the E and the D. In E, if you've got just a single cell, you can inject into the cell. But if you've got more than one cell, then you inject into the yolk sac. So once you've injected this plasmid, the polymerases and ribosomes do their job, and you've got a little programmed CRISPR in there with a DNMT fusion active domain. So this CRISPR is programmed to this little spot. Let's see what it does. You can see, we'll back that up a little bit and go again. You can see that this little CRISPR, with any CPG spot that it came into contact with, it went ahead and methylated. So we actually went ahead and did this. With our proof of concept, we have an Olbus-driven GFP expression in the line of Medaka with a hypomethylated promoter region. So we programmed our CRISPR DCase9 DNMT3A complexes to go target this site and shut down transcription just to see if it was possible. What we saw with a single guide RNA, we achieved a 20% reduction in our GFP expression, and using multiple guide RNAs, we were able to completely eliminate the GFP expression after 6 days in 60% of fish. So very exciting. Where to go from here? There are many candidates for many candidate enzymes that have epigenetic activity. So we need to develop more tools and exploit those other capabilities. We 60% after 6 days GFP knockdown is good. We need to adapt our approach and adjust our approach to achieve a robust response and eliminate off targets. So a little bit of work to do there. And ultimately, we would like to remove and remediate transgenerational epigenetic mutations. if you are exposed to certain chemicals during certain developmental phases in your life, you can you can incur certain epigenetic mutations that exist through generations. Currently, there is no therapy to remediate those effects. So we would like to apply our research um, towards that. Acknowledgements and references. A special thanks to Dr. Ramji Bandari and Dr. Shugang Wong. Without them, I mean, they're both brilliant. Without them, I would not be here. The epigenetics lab has been incredible. The peers, I cannot say enough about Dr. Bandari's lab. And finally, Lloyd International Honors College. Thank you so much. With that, thank you for your time taken.